First of all, do you hear me and can you see my slide?
Yeah,
Yes.
we do.
So I'd like to thank also Professor Esposito and Professor Pession. It's a great pleasure to have the opportunity to share with you our data and also to see you one more time, not in person, but virtually. Nonetheless, it's a great pleasure to see so many friends after so many years. So in this talk, I will cover what we are doing at the National Institute of Allergy and Infectious Diseases in regard to COVID-19. And in particular, I will focus my talk on gene defects and immunopathology of the disease. But I'm happy to take questions at the end on vaccines as well. So here you have a slide that summarizes the situation of COVID-19 2 days ago. It comes from the Johns Hopkins coronavirus website. And as you can see, unfortunately, the United States, as you all know, paid the largest price in terms of cases, but also in terms of deaths. But also it's very sad to see that Italy is also ranking among the first countries in the world in terms of global cases and global deaths. So this is called really for a worldwide effort in terms of research related to understanding the pathophysiology of COVID-19 and eventually attempting forms of treatment and prevention, including the vaccine, which has really gathered investigators from all over the world. And we are only part of them, of course. So what we are doing at the National Institute of Allergy and Infectious Diseases is to investigate COVID-19 in a very comprehensive manner. We're doing this by organizing a network of 15 principal investigators, not just from NIAID, but also from 3 other institutes at the National Institutes of Health, and using state-of-the-art technologies to understand the SARS-CoV-2 pathogenesis. With Alan Su, we've been investigating the genetic basis of susceptibility. We're also looking at resistance to SARS-CoV-2 infection, and this is done in a great collaboration with Jean-Laurent Casanova at the Rockefeller University. We spent some time describing this data. But we're also interested in characterizing inflammation and tissue damage. We do this by looking at biomarkers. The possibility that NETosis may contribute also, so it's a process through which neutrophils may participate in the process. We're analyzing the global transcriptional signature of individual cell types to try to define signatures that mark the pathological response to the virus. We're using proteomics. I will spend some time describing our findings of autoantibodies, which again have been shared also with Jean-Laurent Casanova. We're looking at also adaptive immune responses, in particular analyzing over time development and persistence of specific antibodies to SARS-CoV-2, but also analyzing the T-cell receptor repertoire and whether T-cell signatures of SARS-CoV-2 specificity develop and persist over time in different groups of patients. We're also looking at viral exposure signatures, so to what extent previous exposure to other viruses may shape the response to SARS-CoV-2, the impact of the microbiome with Yasmin Belkaid. And of course, we're interested also in sequencing the viral genome, in particular due to the emergence of variants. We've been doing this for over a year now. So Alan Su and myself, already in February 2020, started a collaboration with several centers in Lombardy, because as you know, Lombardy has been hit very hard and still is hit very hard by COVID-19. So through my, of course, connection with Brescia, we've been working with Ospedali Civili in Brescia, but also with Ospedale San Gerardo in Monza and with the San Matteo, Policlinico San Matteo in Pavia. Later on, yeah, that's are open. So Andrea Lisco and myself, I'm originally from Puglia, so we've also established a connection with 2 centers in southern Italy. As you know, southern Italy in that period of time was not hit as hard, although now there are more cases also everywhere in Italy, actually. And so we have connections with Foggia and Bari. And later on, after the appearance of inflammatory disorders, post-COVID, post-acute COVID in children, the so-called multi-system inflammatory syndrome in childhood, or MSC, we have established collaboration with some pediatric centers in Turin, again in Pavia, in Naples, and also with other centers worldwide, in particular a large collaboration with Santiago in Chile, with Taiwan, with Jerusalem in Israel, other centers in the United States. This has given us the opportunity to investigate thousands of patients with COVID-19 and obtain actually several thousand samples from these individuals. So the first part of my talk will focus on the genetic basis. We're dealing here with a puzzling situation where, as you know, many individuals, once they encounter SARS-CoV-2, remain asymptomatic. They do get infected, but they remain asymptomatic. There are also a significant fraction of individuals who do develop symptoms, but these are mild. And this is obviously mostly true for younger individuals, although there are cases of severe COVID also in younger individuals. Whereas the proportion of individuals who develop severe disease and really life-threatening COVID-19 is much smaller. And it's largely represented, as shown on the right in this slide, by elderly subjects. This is the risk of death per infection according to age groups. And of course, the highest price is paid by individuals over 60, 65 years of age. But this slide also shows the significant importance of comorbidities. Because if you look actually at what happens in the elderly, is that a vast proportion of those individuals who develop fatal COVID-19 also suffers from comorbidities, shown in gray here. And by contrast, if you look at the individuals at younger age, actually the proportion of individuals without comorbidity is significantly higher as compared to what you see in older age. So we have basically two main factors, age on the one hand, and comorbidities on the other hand, that represent obvious risk factors for severe COVID-19. But nonetheless, the fact that actually we do have elderly individuals who get infected and don't develop symptomatic infection or develop mild symptoms on the one hand, these are right here. These are elderly individuals who may even have comorbidities and nonetheless remain resistant to infection or develop very mild disease. And on the other end of the spectrum, we have very young individuals who do not have comorbidities and nonetheless develop very severe COVID-19, made us think that actually there might be genetic factors involved in either susceptibility or resistance to infection. And this has been actually an effort that Helen Su and Jean-Laurent Casanova have been leading for now over a year through an international consortium which is called COVID Human Genome Effort. This is actually the paper that describes how the consortium works and what are the goals of the consortium. So how exactly is this organized? This is a schematic representation of all of the centers in the world that contribute data to the COVID-HG. So each individual basically with COVID-19 that is seen at these centers receives either whole exome or whole genome sequencing, and then all of the data are analyzed together in a joint manner. And we're looking for variants that result in increased susceptibility. There are associated deleterious variants that might be associated with increased susceptibility to COVID-19, or on the other hand, resistance to COVID-19. So in terms of susceptibility, initially, we thought that we would focus on younger individuals, less than 50 years of life. But Helen and myself actually thought that SARS-CoV-2 is a new virus. Yes, there are similarities with seasonal coronaviruses, and also with other coronaviruses that were important for other serious infections like SARS and MERS. But there are specificities to SARS-CoV-2. So it is, to a large extent, a new virus. And as such, even older individuals who have not developed previous histories of severe respiratory tract infections might nonetheless be susceptible to serious SARS-CoV-2 infection if they have underlying gene defects. So we did not restrict our genetic analysis to younger individuals. We took all comers, every single subject that was recruited through our collaboration with Italy and the other sites that I mentioned, received all genome sequencing across the street at Walter Reed, and we analyzed the data and we shared then the data with Jean-Laurent Casanova. And likewise, we did not restrict the analysis to patients that didn't have any comorbidities. We took all comers for the same reason. So let me tell you very briefly give you some introductory slides on what happens following SARS-CoV-2 infection. As you know, SARS-CoV-2 infects the respiratory tract, and this is an example of what happens. This is the coronavirus here, who enters a cell by recognizing a receptor that is expressed on the cell surface. This is the ACE2, and there is also a co-receptor, which is this molecule here, TMPRSS2. So the virus enters the cells, it replicates inside the cells, but at the same time, it induces an innate immune response, which is really not mediated by hematopoietic cells. It's actually mediated initially by the same respiratory tract cells that get infected by the virus. And as a result of this inflammatory response, the cells produce a variety of molecules, including type 1 interferons, also IL-1 beta, and other cytokines that try to contain the viral replication. Now, obviously, if the infection proceeds, then you have a second phase in the infection where you have infection to the lungs, and then this involves recruitment to the lungs of a variety of inflammatory cells, and these are of hematopoietic origins, including monocytes, macrophages, T cells, neutrophils, production of inflammatory cytokines, and this results in a positive feedback, and you have the second wave of the infection. So the first wave is represented by this part of the slide. The second wave in the lungs is represented by this part of the slide. Now, you may have effective immune responses, and again, effective immune responses come also in two flavors. The first one is represented by a strong type 1 interferon response in the respiratory tract, which really is the most efficient way to contain as much as possible the viral replication. And the other one is represented by adaptive immune responses that are elicited in the lungs and throughout the body, in particular CD4 and CD8 cells, and eventually B cells that produce neutralizing antibodies that block the capacity of the virus to bind to ACE2 and to internalize into the cells. But you have many individuals in which these immune responses are dysfunctional. And in fact, what happens in these individuals is that inflammatory responses prevail, and these are mostly mediated by neutrophils, monocyte macrophages, and T cells. And in this setting, you have a hyperinflammatory response that results in serious damage to the lungs as well as to other organs, including the heart, the vessels. As you know, thrombotic events are also very common in this disease. The kidney may be involved, and even the CNS may be affected. So you have two different types of response, and it becomes very important to understand why do some individuals develop effective immune responses and others develop dysfunctional immune responses. So let me again emphasize that what is really important in the very first phase of viral infection, and this is true for virtually any respiratory tract viral infection, is the capacity of the body to synthesize type 1 interferons, and by that mechanism contain the viral replication. So type 1 interferon is produced, in fact, by many cell types. Don't think of type 1 interferon as something that is unique to hematopoietic cells, because that's not true. Although plasmacytoid dendritic cells are the major source of interferon alpha, in fact, type 1 interferon are produced by virtually any cell type, including respiratory epithelial cells, endothelial cells, as well as a variety of hematopoietic cells. So there are examples of other life-threatening viral infections that have been observed in patients that carry rare genetic defects. In particular, we and others have described, and in particular the group of Jean Casanova and Ellen Su have described, patients who developed life-threatening and in some cases, in fact, fatal influenza. And these patients were carrying gene defects shown here, TLR3, RF7, RF9. Ellen Su has also described patients with life-threatening rhinoviral infection, which, as you know, is otherwise a pretty benign virus that causes common cold. But this patient who carried a mutation in this gene called IFH1 encoding for this protein MDA5, in fact, suffered from life-threatening viral infection. What are these genes? These genes are important in response to a viral infection of the respiratory cells are important for producing type 1 or type 3 interferon or amplifying the response to type 1 and type 3 interferon, as in the case of RF9. And this results eventually in induction of so-called interferon-stimulated genes, which are exactly the molecules that help contain the viral replication in the respiratory tract cells. So you have several gene defects that have been previously identified in other viral respiratory tract infections, and we thought that perhaps the same genes might be involved in some cases of life-threatening COVID-19. Indeed, by analyzing several hundreds of DNAs from patients coming from Italy and France, for the most part, again, a collaboration between our group, our Italian colleagues, and the group of Jean-Laurent Casanova and his French colleagues, we identified a variety of gene defects, now shown here in circles in green, that also account for fatal COVID-19. These were all patients that actually required hospitalization in the ICU, and many of them unfortunately passed away. Now, if you look at where are these genes located in exactly the same pathway that I described before, you will see that, in fact, many of them were involved in the production of type 1 and type 3 interferons. Others were involved in a response to type 1 and type 3 interferons. So basically, we have replicated for COVID-19 exactly what had been seen for other life-threatening viral infections. Now, you may ask the question, how common is this situation? Well, we found that at least 3.5% of patients with life-threatening COVID-19 pneumonia carry gene defects that compromise either the production of type 1 interferon or the cellular response to these cytokines. So it's a relatively small proportion because we're dealing only with life-threatening COVID-19, yet it was quite a surprise to find that as many as 3.5% of these patients do carry deleterious gene variants. Now, these variants some of these variants had been previously seen in other patients who had the other type of viral infections that I mentioned before. Other were new variants that had not been functionally tested. And again, these were tested in Jean-Laurent Casanova and Alan Su's lab, and they found actually these variants were loss of function variants. So indeed, they compromised the function of the protein. But at the same time, we reasoned that there might be an alternative way of altering the capacity of the body to respond with type 1 interferon responses. And this might be this might be due to the production of autoantibodies that block these molecules. This is not a new concept. I'm sorry. Do you still see my screen?
Yeah.
Yeah, thank you. So this is not a new concept. There are a variety of autoantibodies that have been described that recognize cytokines.
Gigi, we miss you.
Hold on a second.
We lost your screen.
Okay, let me see how we do this.
Okay.
Thank you. Sorry, let me go back the one slide. Okay, so in other situations, autoantibodies had been described that bind and neutralize various cytokines. In some cases, it had been previously shown that these autoantibodies are disease-causing. I'm going to give you just the two examples. One is autoantibodies against interferon gamma. Patients that do carry these autoantibodies suffer from severe mycobacterial disease, in particular environmental mycobacterial infections. We're going to talk about this in the next class. There are also patients that have autoantibodies that bind and neutralize IL-17 and IL-22. These individuals suffer from chronic mucocutaneous candidiasis. And finally, there are also autoantibodies against GM-CSF, and these are individuals that suffer from pulmonary alveolar proteinosis. So there are a variety of autoantibodies that have been described, and we consider the possibility that perhaps there would be patients with life-threatening COVID-19 who have autoantibodies against the type 1 interferon, in particular interferon alpha or interferon beta, or also interferon omega. So these are Paul Bastard, who works with Jean-Laurent Casanova, and Lindsay Rosen, who works here at NIAID. And so they have screened the serum of patients with, again, life-threatening COVID-19 pneumonia for the presence of autoantibodies against, in this case, interferon alpha-2 and interferon omega. And as you can see here, quite a significant fraction of patients with life-threatening COVID-19 pneumonia had very high levels of these autoantibodies as measured by this fluorescence intensity. And if you look at how those compare to patients who were also infected by SARS-CoV-2 but were either asymptomatic or had mild disease, you see virtually no presence of high levels of autoantibodies in this group of patients, and likewise, healthy controls that were not infected by SARS-CoV-2. These were represented by healthcare workers from Monza who were tested negative for SARS-CoV-2. Again, they remain without these autoantibodies. Now, you have high levels of autoantibodies, but are these autoantibodies blocking the activity of type 1 interferon? So how to test that? So here you have a cell that expresses an interferon alpha receptor, which includes two chains, the interferon alpha receptor 1 and interferon alpha receptor 2 chain. Here you have the type 1 interferon that binds to the receptor, and when this happens, you have activation of the JAK-STAT pathway, phosphorylation of the JAK proteins, phosphorylation of the STAT proteins, including STAT1, dimerization of this protein, forming a complex with RF9 that drives the expression of those interferon-stimulated genes that I mentioned in the previous slide, and these are the ones that help restrain the viral replication. Now, if you have blocking antibodies, not just presence of autoantibodies, but blocking antibodies and neutralize the activity of type 1 interferon, then what happens is that you would not get phosphorylation of the STAT protein, you would not get translocation of this protein, and you would not get expression of interferon-stimulated genes. So we took the plasma of individuals with those high levels of autoantibodies and tested the plasma for the presence of neutralizing activity. So here you have a case where you have a cell that expresses the interferon alpha receptor. You stimulate the cells with interferon alpha in the presence of healthy control plasma. And what happens is that you have very nice phosphorylation of STAT1 just because this plasma does not contain blocking antibodies. Here you have a case of a patient with life-threatening COVID-19 who did have high levels of anti-interferon alpha, but not anti-interferon omega antibodies. These anti-interferon alpha antibodies were not only present at high levels, but they were also neutralizing. They completely blocked the capacity of interferon alpha to induce STAT1 phosphorylation. By contrast, interferon omega normally induces STAT1 phosphorylation because there are no blocking anti-interferon omega antibody. Here I have another patient, also with life-threatening COVID-19, whose plasma contained both anti-interferon alpha and anti-interferon omega neutralizing autoantibodies. In this case, neither interferon alpha nor interferon omega can induce phosphorylation of STAT1 because those interferons are blocked these autoantibodies. If you now take the same plasma and you remove the IgG fraction, so you remove the autoantibodies, then you see that interferon alpha and interferon omega are normally able to induce STAT1 phosphorylation. So this actually showed that in these patients, there were either blocking antibodies to interferon alpha or blocking antibodies to both interferon alpha and interferon omega. So this is what happens basically. You have respiratory tract cells that get infected with SARS-CoV-2, and in normal conditions, the body would induce expression of a large amount of type 1 interferon, which helps contain as much as possible the viral replication. But if you have autoantibodies that block these type 1 interferons, then the viral replication gets unchecked. You have significant spreading of the virus to the respiratory tract, invasions of the lungs, and much higher viral load to the lungs. And eventually, you develop a serious, life-threatening COVID-19 infection. Again, this is an example of what happens in terms of the capacity of cells to get infected, this cell line here, to get infected with SARS-CoV-2. If you add type 1 interferon in the presence of healthy control plasma, basically the type 1 interferon would block the viral replication. You don't see infection in the cells. But if you are now dealing with serum that comes from patients with life-threatening COVID-19, or if you add exogenous anti-interferon alpha-2 monoclonal antibody, then you have a much increased viral replication over time. So again, this is a proof that in a cellular system, the plasma of those patients were basically enabling a much higher rate of viral replication and facilitating development of life-threatening disease. So how common is this condition? Well, we found that as many as 10% of patients with critical COVID-19 pneumonia, you see the number that we tested was almost 1,000 individuals, had very severe COVID pneumonia, were harboring these neutralizing anti-type 1 interferon antibodies. By contrast, we found no cases of neutralizing autoantibodies to type 1 interferon in patients who were asymptomatic or mildly infected, and a very small proportion of individuals who were uninfected and were healthy, but they were uninfected. So this could be potentially at risk of serious COVID-19 if they get exposed to the virus. These autoantibodies recognize mostly interferon alpha and or interferon omega. We found very few patients that harbor neutralizing autoantibodies against interferon beta, and this has important therapeutic implications. We found that these autoantibodies were more common not only among critical patients, but in particular among males and in older individuals. And perhaps that may also contribute to the larger representation of males and elderly individuals among the fatal cases of COVID-19. Finally, as I showed you, they neutralize supraphysiological amounts of type 1 interferon and therefore allowed higher viral replication of SARS-CoV-2, supporting pathogenicity. So we have basically two mechanisms. On the left, you have the genetic defects in the capacity to produce or to respond to type 1 interferon. On the right, you have neutralizing autoantibody, a pre-existing, these are not induced by the viral infection, they are pre-existing neutralizing autoantibodies that facilitate viral replication by interfering with the capacity of the cell to restrict the viral replication through the interferon-mediated process. What are the therapeutic implications of these findings? Well, for patients who harbor genetic defects that lead to impaired production of type 1 interferon, one would argue that the administration of such molecules, at least initially in the course of the disease, is essential to protect them against life-threatening disease. So they don't make interferon themselves, you could provide that interferon. But again, this has been done early in the course of the disease, because as I mentioned, interferons are important in the first few days after SARS-CoV-2 infection. If you wait too long, interferons in a later phase, in the second wave of the disease, actually are pro-inflammatory cytokines. So you don't want to use them in a second phase. You don't want to use them, 10 days, 15 days after the onset of symptoms. You really have to use them very, very early. On the other hand, patients that have neutralizing autoantibodies might benefit from plasmapheresis or from administration of rituximab, other B-cell depleting agents, or anti-CD38, or perhaps proteasome inhibitors to deplete the plasma cells. Importantly, patients that have neutralizing anti-type 1 interferon autoantibodies should be excluded from the list of therapeutic plasma donors. As you may know, the use of therapeutic plasma is controversial, is still controversial, but in any case, if a patient has neutralizing anti-type 1 interferon antibody, for no reason, that patient should be included in a list of plasma donors, because that might aggravate the disease phenotype in the recipient. Now, in the second part of my talk, I want to talk about inflammation of COVID-19 and the work that we have been doing. And the questions we ask are the following. What is the impact of the infection on inflammatory biomarkers? How do the levels in the blood of these inflammatory biomarkers correlate with the disease severity? And in particular, can we identify biomarkers that are predictive of poor or favorable outcome? We've done this in multiple ways. One option was to use this technique, which is called site-seq, where you basically label the cells with antibodies, and these antibodies are barcoded. And then basically on a single cell level, you analyze the transcriptional profile. And because you have these antibodies, you know exactly which cell you're dealing with. So you obtain basically a single cell RNA sequencing analysis, and you are able to identify what is the transcriptional profile of the patient in a given cell population at that given time in the course of the disease. This study has been done by John Sang. The study has been recently published in Cell. So one important point to consider when one does this kind of studies, and that applies also to soluble biomarkers, is that you have two main variables that you need to take into account. One is the disease severity. It may be very different if you're studying a patient with a mild disease versus a patient with a severe disease. But also, it's very important to understand the importance of the time since symptom onset, because these biomarkers may fluctuate over time, and may fluctuate in a different manner in patients that have mild disease versus patients that have severe disease. So both factors have to be taken into account from a bioinformatics standpoint when analyzing the results of the study. So this is the Cell paper, and basically what John found was that by analyzing blood cells from these individuals at various time points, individuals who had severe disease or more modest disease, he found that there were signatures of apoptosis in plasmacytoid dendritic cells. Now, this is important because, as I mentioned, PDCs are the main source of type 1 interferon in the blood. So if you find that these PDCs are dying, that would be another reason to have low levels of type 1 interferon, irrespective of genetic defects, irrespective of autoantibodies. In this case, it's a virus infection itself that induces death in the PDCs, and by that mechanism reduces the capacity of the body to produce type 1 interferon. We also found that there are metabolic abnormalities in natural killer cells, which is another cell type of the innate immune system that fights against viral infections. We found that there were time-dependent changes in several inflammatory biomarkers and in inflammatory cells, and there were trajectories of soluble biomarkers, in particular, that diverge around day 20 since the symptom onset, making patients who died very different than patients that recovered. So this time point is and we all know this. We all know that around day 15, day 20 since symptom onset, day 15 since admission, this is a time where actually you can unfortunately say this patient is really, unfortunately taking a bad trajectory, is at high risk of dying. This is a critical time in the course of the disease. This is one example of what happens in response to interferon signature. You can see here that for patients with mild disease, they start with a very high type 1 interferon response, which is actually what you want. But then this type 1 interferon response goes down over time. You don't want to have a high type 1 interferon response later on in the course of the disease because, as I mentioned, at this phase, type 1 interferon is pro-inflammatory. But on the other hand, patients who actually develop life-threatening disease, they started with a much lower type 1 interferon response, unable to restrict the viral replication, and actually they developed a much stronger type 1 interferon response later in the course of the viral infection. So you have basically a dichotomy in the type 1 interferon response trajectory between patients with mild disease and those with life-threatening disease. In another study that we did and we published in JCI Insight, we analyzed 66 soluble biomarkers in 609 blood samples from 117 patients with COVID-19, all comers again, from asymptomatic to fatal cases of COVID-19. We identified 12 biomarkers for which, even at the first time that the patients were sampled after admission, these biomarkers were able to distinguish those patients that were at higher risk of dying from those that actually that survived. So this is the adjusted hazard ratio for death, and you have here the list of these biomarkers that were associated with increased risk of death. And 4 of them, at any time point in the course of hospitalization, clearly distinguish the patients who died, shown here in red, versus those that survived, shown here in blue. So we think that these are valuable biomarkers because one could use these biomarkers and actually identify which patients are high risk dying of the disease. And these are patients that will require very aggressive treatment already from the very beginning. As you know, the strategy normally is to use dexamethasone, in some cases methylprednisolone, in patients who develop severe pneumonia. But perhaps in patients that actually have this inflammatory signature, one should be considering using more aggressive form of treatments, maybe including dexamethasone, JAK inhibitors, tocilizumab, BTK inhibitors, already early in the course after admission, because these are patients that are at very high risk of dying. So we think there is value in using these biomarkers. Finally, let me tell you something about the adaptive immune response. The adaptive immune response is developed later in the course of the disease, but they are very important because they protect us from reinfection. And this is actually, this is a strategy also the vaccines are using in trying to induce an adaptive immune response in order to prevent infection in the individual. So which adaptive cells are we talking about? We're talking about 3 different cell types: B lymphocytes, CD4 positive T cells, CD8 positive T cells. This is the coronavirus, and shown here are the various proteins that characterize the coronavirus. Very important are the spike protein, shown here. Then you have the nucleocapsid and the membrane protein, shown here. As you see, the spike protein interacts with ACE2 and also with this other co-receptor TMPRSS2, and that actually helps the virus enter the cell. So CD4 positive cells are crucial for both B cell and cytotoxic T cell responses. On the one hand, they elaborate cytokines that activate the B cells and enable the B cells produce antibodies that bind to various proteins of the coronavirus, including neutralizing antibodies that bind to the spike protein and impede recognition of the ACE2 protein and bind to the ACE2 protein, and therefore impede internalization of the virus. But CD4 positive T cells also elaborate cytokines that activate CD8 positive T cells, and these CD8 positive T cells, in turn, they produce cytolytic proteins like granzyme that allow killing of the virus-infected cells, and by that mechanism also clear the viral infection. So we've been interested in understanding how good are these immune responses after SARS-CoV-2 infection in some individuals who are at particularly high risk of developing severe COVID-19. And again, these are individuals of age over 65 years. We're interested in patients with hematological malignancies because, again, they are at high risk of severe COVID-19, or also patients who were on heavy immunosuppressive treatment, perhaps because they were recipients of solid organ transplantation or had other conditions of immune compromise. So let me show you the data. These are the data of antibody responses up to 6 months and over after the initial infections. On the top, you have patients that do not have hematological malignancies. On the bottom, you have the antibody responses in patients with hematological malignancies. 2 types of antibodies: the anti-S antibodies, which include the neutralizing antibodies against the SARS-CoV-2, and these are the anti-nucleocapsid or anti-N antibodies. So the good news is that in the vast majority of individuals that got infected with COVID-19 and didn't have hematological malignancies, very high levels of antibodies are produced relatively soon, and they persist over time. We really don't see much of a decline of these antibody titers for up to 6 months. Very rarely you have individuals who fail to make good antibody responses. And these are mostly individuals who had asymptomatic or mild disease. But if you look at what happens in patients with hematological malignancies, you will now appreciate that the proportion of patients who fail to make protective antibodies, the gray area is basically non-protective, and everything above the gray area is protective titers of antibodies. You see here, basically, these individuals fail to make protective antibodies, and some do make antibodies, but they decline rapidly. For anti-N, again, you have lack of antibody production in a significant proportion of patients with hematological malignancy, and you see the decline in many other individuals. So clearly, patients with hematological malignancies do have problems in mounting antibody responses. This is partly due to the fact that some of these patients had received rituximab as part of their treatment, mostly for non-Hodgkin or follicular lymphoma. So this makes interrogating at this point the other arm of immunity, which is the T-cell immunity, particularly important in this group of individuals. How good is the T-cell immunity in patients with hematological malignancies? How durable is the T-cell immunity? And of course, this has also implication for immunization of this group of patients. So T-cells. T-cells, of course, recognize peptides, including the SARS-CoV-2 peptide antigens, in the context of HLA molecules or MHC molecules. You have two different types of T cells. You have CD8 positive cells that recognize peptides presented in the context of HLA class 1 molecules. But you have CD4 positive cells that recognize peptides in the context of HLA class 2 molecules. These are the CD4 positive cells. These are the CD8 positive cells. So in all cases, recognition of the peptide is due to the specificity of the T cell receptor. And so one thing that we have been doing has been from the very beginning to collaborate with Adaptive Biotechnologies and Microsoft, trying to, in a bioinformatic approach, to identify specific sequences of the T cell receptor that identify T cells and recognize SARS-CoV-2 epitopes. This has been facilitated also by an in vitro assay called MIRA, in which cells have been stimulated with peptides, and the T cells are responsive to SARS-CoV-2 peptides are selected, and their T cell repertoire is sequenced so that we know for sure that T cell receptor is specific for the SARS-CoV-2. But then from there, we moved on, and without doing any in vitro cell assay, we just sequenced by high throughput sequencing the T cell receptor repertoire, and we try to identify private and public sequencing of the T cell receptors that are specific for SARS-CoV-2. These are two papers that have been published, one by our group in collaboration with the Adaptive Biotechnology group, but the other paper is also very interesting. You may know the story, of course, of Vo in Italy, and in particular, you may be familiar with Andrea Crisanti. So as you know, what he did was basically to sequence all individuals that live in this village, Vo, and he analyzed the T cell receptor repertoire of those individuals to identify exactly which patients had been exposed to SARS-CoV-2 and for how long the T cells of those individuals persisted in response to SARS-CoV-2 infection. So I recommend reading that paper. So again, the T cell receptor is generated by recombining variable diversity and joining genes that are scattered along the T cell receptor loci. And in particular, this allows formation of what are called complementarity determining regions, 3 of them. Now, CDR1 and CDR2 establish contact with the MHC, with the HLA molecules, but the CDR3, which is the result actually of the joining exactly of the V, the D, and the J elements, this is the region of the T cell receptor that establish contacts with the peptide. So the specificity is really contained within the CDR3 region of the T cell receptor. And that's exactly the region that we sequence. And so if you look at SARS-CoV-2 epitopes, amino acid sequences shared by the spike protein here, in individuals who have this particular HLA typing, they carry this allele A02, you will see that there are some peptide residues that are recognized by T cell receptor alpha or beta chains. So one should really focus on restriction dictated by the HLA typing and look at now at the sequence of the T cell receptor that binds these peptides. So we did that, and we realized that actually that specific sequence may be even coming from different V and J genes. So you have this process that is called convergent recombination, whereby even if you rearrange different V and J genes, nonetheless, you're able to generate the same CDR3 sequence, and so you're able to bind exactly the same epitope of the SARS-CoV-2 protein. It's an important concept. Now, when we looked at individuals who were A2 positive, and we separated those that had mild disease, moderate disease, severe disease, and critical COVID-19, you can clearly see here that there was a signal that was shared by many individuals. So these are so-called public clonotypes. They identify sequences of the CDR3 that are present in many individuals. So it's the same sequence that is shared, and for that reason, it's called a public clonotype. Now, the problem was that when we analyzed healthy controls, we also saw that the same signal was present also in healthy, uninfected controls. So how come that you have healthy control T cells that are capable of recognizing the same SARS-CoV-2 peptide that patients who got infected with COVID-19 also did? Well, the reason relies on the presence of cross-reactive T cells against other coronaviruses. This is a phylogenetic tree of coronaviruses. This is actually the SARS-CoV-2 virus, named here from the Wuhan U1, which is the original name that was given. This is a beta coronavirus, and you see there are other two well-known beta coronaviruses that were responsible for serious disease, SARS and MERS. But you have also common cold coronaviruses. These are responsible for much milder diseases in humans, and some of them are beta coronaviruses, shown here, and others are alpha coronaviruses. Now, these coronaviruses share some similarity with SARS-CoV-2, and that may explain, in fact, why you have T cells that may recognize SARS-CoV-2, even in individuals that have a previous history of infection with these seasonal coronaviruses. So, in fact, if you look now at the frequency, though, of SARS-CoV-2 specific T cells in healthy individuals, pre-COVID era, or individuals who had other kinds of viral infections like CMV or EBV, again, pre-COVID, you will see that there is a low frequency of SARS-CoV-2 specific T cells. But the frequency is higher in individuals who actually got infected with COVID-19, and even more so in patients that develop critical disease. So the total frequency of T cells that recognize SARS-CoV-2 tends to be higher in individuals who develop more severe disease versus those that develop mild disease, and in any case, much higher than individuals who were not infected and perhaps had just cross-reactive T cells. Now, if you look at age and you analyze and you ask the question, how diverse is the capacity of the T cells to recognize epitopes of SARS-CoV-2? What you can see here is that individuals who are over 65 actually recognize much fewer epitopes of SARS-CoV-2 than individuals who are younger. So even if they can recognize SARS-CoV-2, their immune system is not able to recognize as many parts of the virus as in younger individuals. Their T cell responses are less good than in younger individuals. They have a more narrow repertoire of T cells against the virus. And if you look at patients with hematological malignancies, you see pretty much the same. This is the repertoire of 13 patients with hematological malignancies, very restricted in terms of their capacity to recognize SARS-CoV-2 as compared to patients of the same age but without hematological malignancies. This is not specific to SARS-CoV-2. If you ask the question against any other antigen, influenza, whatever, you will find exactly the same thing. So patients with hematological malignancies, because they received chemotherapy, because their thymus was damaged, because there is senescence in the immune system, their repertoire is not as good. So let me conclude by saying that in the second part, I've described basically inflammatory biomarkers that are predictive of poor outcome, even when measured early in the course of the disease. And this may help perhaps use more aggressive form of treatment for these patients. I've shown you that specific antibody responses persist for more than 6 months in infected subjects. However, these responses are not as good in patients with hematological malignancies. These patients, as well as patients of older age, have reduced diversity of their T cells against SARS-CoV-2. And I think these are important information to take into account when considering immunization policies for this group of subjects. It will be important to see how good their antibody responses will be, how good their T cell responses will be. And we're actually doing such studies as I speak now. Lastly, let me finish by encouraging everybody to get immunized. This is an old slide, and it's actually taken here from the United States. This is actually a grocery store, but in this case, it says, for getting food entered through the other door, this door is just for polio vaccination. They were using any venue to immunize individuals. I hope that this becomes true more and more in the United States and definitely in Italy. I know how bad the situation is in my country, and I hope more and more people can get immunized as soon as possible. Lastly, I want to emphasize the many people who helped do this work. Again, a great collaboration with Sharon Casanova and his team, and Ellen Su, people from my lab, Kerry Dobbs, Ottavia Del Monte, the bioinformatic that works with me for the T cell receptor analysis, Jiang Dogus, collaborators at the NIH, but also people from Italy, in particular from Brescia, Monza, and Pavia, who have made it possible, and our collaborators in the hematology unit in Brescia, with whom we're working for the hematological malignancies.